Yeah, it just started good. I know, I parked it dry. Yeah. This is the. It's Orlando, right? No, it's Houston. You know that thing that's probably me. Still on the list?
Yeah.
Yeah.
Bye-bye. I guess we're back to
How many?
Yeah, was that when it goes original?
Yeah.
Go to the next please. Next slide, yeah. Yeah.
So that's good. Just fried breakfast. Probably don't want to see. There's some terrible federal traffic. Okay.
Hello. Hello. You all same. Same. Oh, you're right.
It comes up once a year. Yeah. Since last year.
We're going to walk. Parking lot. It's done. Oh, are you? Okay. All right.
Go back. Driving around in their valley.
You know,
They're running to the ground. You don't see them.
What my head was that last day?
Oh, that.
Ridiculous.
Oh, yeah.
Yes.
Oh, yeah. And everybody has.
Yeah.
Can't be somebody that disappears. The women were really bad.
Yeah, I parked in the place too early to get a ticket after 11 o'clock p.m. And I'd gotten a ticket there once before. 11 to 9. So you can either put this or I left it before the, order back. But I'm sure it was impressive. Did you see the little drawings for the hell?
Oh, yeah.
It's amazing what kind of trees we got. You don't get that perception, you don't get that. I don't understand why.
Everything's fantastic, but all.
Yeah, they do a good job with that. Is it CNBC?
And then it's like, take us all out to the bar. Like, everybody drinks. My friends loved it. They like dabbed me and they got.
Oh, yeah.
Yeah, you gotta spread the love, you know. So it's all about. But buy some grapes. Get ready.
Oh, I'll buy some. I'm like, yeah.
Pictures of Bush Light. $5. Yeah, I know. I've done that.
Mike, can you run there? Sure. So yesterday,
So I ran into
What about kitchen?
Yeah, but I talked with my
Oh,
I'll be there this month. That's why
yeah. Yeah,
otherwise it was.
Yes, we didn't know what was going on.
Yeah, okay. I just wanted to.
Yes.
Because I was like, and never told it. He just had to
Did you check? Confirm the address?
Yeah, right. The friends we did building in. Not that I sent them were accurate.
The word of the day is Evanston.
I'm not sure that anything else.
Yeah, that was it.
Yeah, so
My umbrella was open.
Mine was having a good time.
No, they're cheap. You can just buy a new one. Maybe I'll get you one for Christmas.
Ellis Dow. Okay, it's having it, whatever.
Let's go ahead and get started. For those on the outside, if you didn't hear it through, the word of the day is Evanston. I don't know what that.
Evanston.
Evanston, like a city outside of Chicago.
Oh, for Northwestern,
Oh,
the city.
did you train at Northwestern? What you're saying? There you go.
That's actually
All right. So Dr. Singh from our Pulmonary department, he's actually the first speaker, local speaker. Everybody else so far this year has been visiting firemen. So thank you for coming. Hopefully his presentation is not going to be compromised. He wanted to do it on a Mac, and usual technical problems that couldn't get the Mac to work. So this is on more or less on biofilms, or why bacterial sinus infections are so hard to cure. Thanks for coming.
Thanks a lot for the invitation. I appreciate it. I just want to clarify that the Mac works fine. It was the PC system. So I'm going to talk to you about some new ideas that we and others have been working on to try to understand why chronic infections are so difficult to treat. And so I'm joint between the medicine and the micro department. We kind of view ourselves as these professor of barbarian studies that are following around by barbarians trying to understand what they're up to. The barbarians are the bacteria, and they're a lot smarter than we think. So I'm going to start out with a little case, which is probably more familiar to you than to me, but just to illustrate this point. So this is a patient with chronic sinusitis, quite young, recurrent purulent rhinorrhea, treated with a kind of a standard course of antibiotics, augmented for 3 weeks, saw some improvement, but then his symptoms came back right away 2 weeks later. Got an allergy immunology consult, and no triggers were identified. Cultured and grew pan-sensitive Pseudomonas aeruginosa, and then was prescribed 6 weeks of levofloxacin and got some improvement. But then a month later, the symptoms came back, and the culture results, interestingly, were unchanged. So the same organism with the same sensitivity. And since then, this patient's had multiple flares like this and eventually got referred to surgery. And so this is a common problem that you face, I think, and that we all face in medicine. And it's a problem really about chronic infections. Now, I want to just show you that you're not alone in dealing with this problem. So this is an example of treating cystic fibrosis infections which also caused by Pseudomonas aeruginosa with high doses of inhaled tobramycin. And what's plotted on the y-axis is the bacterial counts in a log scale. X-axis is time. And the shaded areas are where the antibiotics were administered. And what you see is that the antibiotics work. They reduce the bacterial counts by 2 logs, so 99% of the bugs are eliminated or killed. But then as soon as the antibiotic is stopped, the counts come back, and this cycle is repeated over and over again. So this is a chronic issue with chronic infections. Here's another example: antibiotic therapy for infectious kidney stones. You see the same general pattern. Antibiotics work, suppress the infection, lower the counts. As soon as the drug is removed, the bacteria counts balloon. So So the questions that I want to address are really, why are these chronic infections of all types, chronic bacterial infections, or most types, so difficult to eradicate with conventional antibiotics? And the first point I want to make is that it's not, I don't think that the major issue relates to what we normally think about as resistance, fixed resistance caused by genetic mutations, at least in the conventional sense. And let me show you an experiment done by some of our colleagues at Children's Hospital here at UW. This is in cystic fibrosis. And so what's plotted on the y-axis is the change in lung function of a CF patient treated with antibiotics. And on the x-axis is plotted the tobramycin MIC of that person's isolate, right? So what you would expect is some kind of relationship like that. If they had a very sensitive isolate, you'd see a big change in lung function, big improvement in lung function. If they had a resistant isolate, you'd expect to see a less of a change in lung function, less of a response. But what you actually see is something like this, with no clear relationship between the bacterial MIC, how much it takes to kill the bug in the lab, and the patient's response. So that really tells us, I think, and other work suggests that there's other things going on that we need to start thinking about as a cause for this treatment resistance of chronic infections. So I'm going to tell you about two basic ideas today about that we've been working on in cystic fibrosis, chronic bacterial infections, which I think are probably generally applicable, that might shed some light on this phenotypic treatment resistance phenomenon. And so one of them is this notion about genetic diversity. And so genetic diversity evolves in bacterial populations and can increase the resistance. So let me show you what I mean. So in CF, we have kind of a steroid, chronic infection on steroids, which probably bad choice of words in this audience, but basically what happens is the patient's born with sterile lungs, an infecting Pseudomonas gets in their lungs, and then it stays there for 20 or 30 years. That lineage of bacteria, the progeny of this infecting isolate, stays in the patient's lungs, eventually causes inflammation and respiratory failure and death. So all the bugs that are there chronically are the progeny of this infecting strain. Now they have to evolve inside the host. They're replicating like mad. A lot of them are being expelled from expectoration, but yet the population stays generally constant. So that means a lot of replications occurring. That means evolution within the host must be happening. And so we wanted to start exploring how evolution of the bacteria inside the host might occur. And there's two general patterns that could occur, and these are oversimplified, but I think it helps illustrate the point. So one possibility is that within host evolution of the bacteria during chronic infections could produce a dominant strain that's adapted for that lung environment condition, or in your case, maybe sinus environment condition. So here's how that might work. You have your initial strain, it starts replicating, genetic replication errors occur, and genetic variants arise. That's indicated by the star here. And now you have two clonally related strains, but one of them is particularly fit for the lung conditions. So it goes on to propagate. You get again, and genetic mutations occur. One of them is more fit, and so on and so on. And at the end of this process, you have a strain that's different from the initial colonizer. It's a sibling, okay, genetically related, but it's adapted for the lung environment. So that's one general possibility. The second general possibility is that we have our initial infecting strain. Same process occurs with genetic variants being produced by mutations or replication errors of DNA. But now more than one isolate stays in the lung. And selective conditions in the lung or the chronic infection source, wherever it is, favors more than one. There are different niches where both can survive simultaneously. And you can imagine that going on and on and on. And then at the end of this process of evolution, what you have is a diverse zoo, okay, community of clonally related siblings that populate that infection site. And we think these two possibilities are important to distinguish because they have very different implications for infection. So here's a very simple experiment done by a fellow in my lab, Ben Staudinger. And so he wanted to ask, well, okay, we know we got these CF patients. We know that they're chronically infected with Pseudomonas. We know that the lineage is constant. So the bugs in the patient's lungs a decade after infection are related to the initial strain. Let's collect sputum, and instead of doing what the clinical lab normally does, which is plate it out, pick a colony, okay, one bug, one colony representing one cell, and saying that's your infection. He picked 1,000 or actually 1,500 different colonies. He subjected them to a number of phenotypic tests after passaging the bugs. So these are heritable phenotypes to ask the question about how much genetic diversity existed in this bacterial population. Now, let me just show you some examples of what he found. But first off, I don't have time to show you this data, and it's not really that relevant, but I want to remake this point that all these isolates that he analyzed are genetically related to the clone that started the infection. They're all siblings. And so the first test he did was he said, well, let's see if they can grow in the presence or absence of amino acids. And so what you're seeing here, this is a plate, doesn't have, what happened?
Back rib.
Yeah.
Drew, there's a resume slideshow button in the middle there. Which one is it? You just hit the
So it knows it did it. I guess awareness is the first step of. So he plated this on a bacterial plate that lacked amino acids. So what you're looking at is arrayed colonies. These are all in a row. There should be 96 of them here. They're all from the same patient, and they're plated on this plate that doesn't contain any amino acids. And as you can see, some of these siblings can grow very well without amino acids. Some of them can't. They don't even show up on the array. So that got him excited. He started looking. I don't need that. Started doing another test. This is a swimming motility test. Let me show you what it looks like. He punches down an isolate from the patient's collection that's living in their lung, simultaneously present in the lung, puts them in semi-solid agar. If they can't swim, they stay in a ball. If they can swim, the whole well becomes cloudy. And what you see is that there's an amazing amount of diversity in the capacity of these siblings to perform swimming. Some of them swim really well, some of them don't swim at all. Here's another test, getting more clinically relevant. What about if we plate these on antibiotics? What about their antibiotic susceptibility? Quite a bit of divergence in antibiotic susceptibility to ciprofloxacin. Some grow real well, some don't grow at all. Here's another example. High doses of tobramycin. Some isolates in the population grow, some of them don't. So that got him very interested and us very interested, but it also drove the point home that this is really going to be complicated because we're seeing this amazing diversity, the dramatic diversification of the bacteria in vivo in clinically relevant traits. Okay? And so the first thing that Ben decided to do then was to look at this and say, well, how can we make some sense out of it? So he devised a classification scheme. So let me walk through this with you for a second. So what you're looking at is one isolate subjected to multiple tests. Okay, this is all the same isolate. If it produces this surfactant called rhamnolipid, it has this kind of a color. If it can grow on amino acids, you can see a colony. If it doesn't swim, it stays there. If it doesn't grow on tobramycin, etc. This is a phenotyping of one colony out of the 1,500 that he picked that coexisted in that person at one time.
How many divisions do you need to have such diversity? Do you have any idea how many times they replicate?
No, I don't have any idea. I think that's very, very complicated, right? Because we have no way to measure what the population, how much is lost every day, and we have no way of measuring what the population size is. We know the concentration of bugs in the sputum. It's about 10 to the 8th or 10 to the 9th CFUs per mil, but we don't know how many mils, and we don't know how much are eliminated every day, and we don't know the growth. But I would say a lot. A lot of replications. So back to this. So here's the phenotype of one colony. It has this particular code of phenotypes, and if it had that code, he assigned it to do a subpopulation. He said, everybody that's got that phenotype, heritable phenotype, let's assign it a number 1. If it has something different, let's assign it a 2 or 3 or 4. And what he found then, in every chronically infected patient that we've examined is a pattern that looks like this, where there are, on the y-axis here is plotted the percentage of the total population. This is all colon-related Pseudomonas. On the x-axis is plotted these arbitrary binned subpopulations defined by this exercise up here. And what he found was that in every patient there were 1 or 2 or sometimes 3 highly dominant variants. And then there was a very long tail of rare variants going out here. And we think we're just scratching the surface because every time we add another test to this, the tail gets longer and we see more and more variants arise. And however, we're kind of limited by the number of, this is 1,500 isolates from one sputum sample. We're limited by a test that we can do in high throughput manner. Otherwise, out of business. And so, but we see this pattern. And I want to just call your attention to these rare populations because one might easily dismiss them and say, well, geez, this is present at I don't know what it is, but 0.1% of the population at any given time. How important could that be? Well, I think it could be very important, and I'll get to you explain that why. So we see this quite amazing, dramatic population diversification of a single clone inside patients with CF. So how is that going to affect disease manifestation? How might it affect disease manifestation? Well, the ecologists have been thinking about this relationship between diversity in populations and stability or resilience of those populations for a long time. And there's a notion in ecology called the insurance hypothesis. Insurance hypothesis says that the presence of subpopulations with diverse phenotypes extends the range of conditions in which the population will thrive. Okay? Diversity in a population makes the population more resistant and resilient. And so how might that work during a chronic infection? Well, one possibility is, I made a little cartoon here to illustrate some possibilities. So one possibility, here's when our patient's well. On the y-axis is the total lung counts, and each one of these different colored bars represent a different clonal variant that's present in that population. You got a dominant one, you got some rare ones. And so one possibility is that these minor subpopulations have evolved to use nutrients not favored by the major subpopulations, to exploit unexplored niches or unexploited niches in the lung. And if that's true, that this phenomenon could increase the total population size, because now you have variants that can use different nutrients that if there wasn't that diversity, those nutrients would go unused. So that's one potential way that diversity could increase or have an effect on infection. The second way is illustrated by this bar. I come along, I put my patient on antibiotics, and as I showed you from those initial examples, antibiotics suppress the population quite well. But what's going on with the subpopulations? So maybe what happens is that I effectively suppress many different subpopulations, but there are rare variants in the population that can handle or are resistant to whatever antibiotic I might throw up. So they increase in abundance and compensate for the loss of the others, and therefore blunt the effect of antibiotic treatment. So that's how the insurance hypothesis might work in infections. So let's see if we can test this. Now this is tricky, this is hard, and I don't think we've tested this adequately, but we're starting to work on it. So when you think about what the insurance hypothesis demands, or the biological insurance idea demands, first of all, the diversity in the population needs to affect phenotypes that are important for survival in that environment. If it's just if it's just eye color, it's a heritable phenotype, but it doesn't matter. It doesn't affect survival capacity or fitness. So we need to know that this diversity affects phenotypes important for survival. And secondly, we got to be able to show that population composition changes. Thanks. When adverse conditions arise in the lungs or to the population. And that will also is a part of proving this notion of insurance. That's okay with me if it's all right. So, yeah, it's okay. So could it be that this diversity is improving the fitness of Pseudomonas inside cystic fibrosis lungs? Well, the first question is does the diversity affect traits that are important? And the problem is that we don't know what traits are important in the lung. That's kind of speculative. We don't really know what conditions the bacteria are living under and what are the key stresses that they face. And so I want to show you an experiment where we try to take an unbiased approach to address this question. And so what we did was we used the technology called phenotypic microarrays. And basically there are a bunch of plates, 96 well plates, 2,000 different wells altogether or so, in which every well has a different stress or nutrient condition, okay? And then what we did was we said, well, let's compare the most abundant isolate that exists in a particular patient's sputum to the pool of isolates that coexist to ask how the pooling or the diversity in the pool might affect the functioning of the bacteria. And so here's the experiment. Take the abundant isolate, put it in a stress or nutrient condition, compare it to what the pool that exists there in the lung does, and then I get a readout. It's basically a function of growth. Y-axis is a growth measurement, x-axis is time. Here's here's what the pool does, and when I overlay them, I get a graph like this, where yellow is they both did where the red and green overlap, and the red here showed enhanced fitness. And so this is the data that this kind of an experiment generates. And so this plate contains a bunch of different carbon sources that bacteria grow, a bunch of different nitrogen sources, phosphorus and sulfur sources, antibiotic stress conditions. And what you see is a lot of red, okay, which says that the pool, in all those conditions where you see red, the pool functioned much better and grew much better than the individual isolates. And so I'll show you a few examples. Here's growth on citrate. See, the pool did a lot better. Here's growth on arginine. I can keep showing you these. There's no norfloxacin, streptomycin. You see this conserved pattern where the diversity markedly increases the capacity of the bugs to use nutrients and resist stress. So the second point about insurance, biological insurance, is that I should be able to see the relative abundance of subpopulations change when I apply a stress. And in this case, we're applying the treatment stress of antibiotics. And so here's the graph I showed you, or a similar graph I already showed you about the population diversity in a patient before treatment. Come along, put the person on antibiotics, and this is what I see. I see that some of the very abundant isolates go down in relative abundance, but then rare variants pop up and have the effect of compensating for the loss of the more abundant isolates. And so the idea then is that this provides assurance because the rise in these subpopulations compensate and sustain infection during treatment.
What's the driving force for the diversity to develop? Theoretically, the initial bugs, all the bugs are growing in the same environment. Why does some change their genes and phenotype?
That's a great question. I think that, the answer is I don't know. But we have some ideas about that. And I think it has a lot to do with geographic heterogeneity and geographic isolation of populations. So if for example, I have a group of beetles and a river arises and separates that group of beetles so that they're no longer in competition with each other because they can't get across a river. And they're living under different environmental conditions, eventually they're turned into different beetles, right? It's like speciation. And I think the same thing happens in the lung. And I think the same thing happens in the sinuses, where you have a bug in one environment taking one evolutionary trajectory. It's not in direct competition with somebody else because of physical separation, and it takes a different trajectory. So this insurance is daunting, but I think it also provides opportunities for us to try to understand what causes treatment resistance in a human. And so I'm going to just show you an example of that. This is just the first 6 subpopulations of this graph that I already showed you. This is what happened before and after treatment. Subpopulation 5 bloomed up. And so what we started to do is to say, okay, let's compare 1 and 5. Let's compare representatives from the group 1 and representatives from the group 5, see how they differ, and maybe that could tell us what functions in the bugs are important for treatment resistance in the human. Let's use them as canaries in the mine shaft. And so here's an example. And this is work done with Jim Bruce, actually here down at South Lake Union. And what you're looking at is proteomic data. Here's a drug efflux pump. Here's population 1. It expresses at low levels. Population 5 expressed it at high levels. That makes sense. That would make this guy resistant to treatment in vivo. But here's some other genes which don't even have annotated functions yet. They're hypothetical. We don't even know what they do. And note this y-axis here. Here I saw a 3-fold increase. Here in the resistant population in vivo, I saw 30 and 90-fold approximate increases. So that tells us or suggests that there's these bunch of unknown functions in the bacteria that could be mediating in vivo treatment resistance, and we need to try to figure what those things out. So to summarize what I told you so far, evolved diversity probably increases the capacity of the infecting population to use nutrients and resist stress. Population composition changes during treatment, and subpopulations may compensate for each other to maintain biomass, therefore maintaining treatment. And so I guess the take-home message is that what we've been doing in treating chronic infections is that we've been playing whack-a-mole. We apply a stress, some things go down, other guys pop up. And that's a consequence of the evolved diversity. And instead of playing whack-a-mole, we need to start playing chess and figure out how we can devise better strategies to account for this population diversity.
Question.
Yeah.
If you go back and look 6 months after treatment,
yes.
they return back to subpopulation 1 largely, or?
Well, I really want to know the answer to that question. And our preliminary data suggests that maybe that happens. We've had a lot of trouble getting full collection sets from people, sick pre-antibiotic, on antibiotic, and then returned to well. It's a clinical research challenge. We have a few sets like that, and the data suggests that what you're saying.
Well, you think so, since you said most people start with that largely 1 and 2, and I assume a lot of those patients receive various antibiotics.
Yeah, well, I would just point out that 1 and 2 are arbitrary. Those are just bin.
Right, but it's the same 1 and 2.
No, it's not the same 1 and 2 for 1 and 2 is just 1 is the most abundant, 2 is the second most abundant. It's just arbitrary. We haven't found a clear association between what you're saying, phenotype and patient. That's a good question. So I just want to point out to you that this is not necessarily new phenomenon or understanding. The cancer docs have understood this for some time. And so, and that's the idea of this shifting abundance of clonal variants. And so here's an example in multiple myeloma. Okay, the patient, these are also all clonally related variants, right? They're derived from the patient's cells, and they phenotype them into different subpopulations, different genetic variant populations. This is the most dominant. This is less dominant. Then they put the patient on treatment. Red guy goes down, green guy pops up. They change the treatment, green guy goes down, and this brown guy pops up. And this is known now in cancer. And this is probably why, one of the major reasons why cancer is so difficult to treat. Because you're not treating one cell type, you're treating a zoo of genetic variants with different functionalities. And I think the same thing's going on in chronic infection. So let me tell you about the second point I wanted to make, or second idea about why bugs. Before I leave that, I just want to tell you that I didn't have time to show you that data, and it's not as well developed. But we started looking at this in chronic sinusitis with Greg Davis at UW, and we see a similar phenomenon. We haven't looked at the change in treatment yet. We just looked at stable populations. But we see the Pseudomonas in a chronic sinus infection have also diversified in similar. So the second point I'd like to discuss is this idea about another idea about why the treatment is so difficult or so inadequate for chronic infections. And I think it's because the bacteria are growing in many chronic infections rather than as free-living organisms. They're growing in these aggregates. And these are highly resistant to killing. And so this idea has kind of come out of the biofilm literature. And we now kind of think about the lifestyle of bacteria in two different ways. You can imagine them as free-living independent cells suspended in a liquid broth medium where each guy is his own operator, or you can see them growing in clusters or aggregates. Some people call these biofilms. I'm not sure that what biofilm means anymore because it's used in many different ways. But what you generally see is this aggregated growth mode. That's what you see in chronic infections. And let me show you some examples of this. So here's a bunch of bacterial aggregates on the sinus mucosa. You can see these are individual rod-shaped bugs, probably Pseudomonas, encased in a matrix growing on the sinus mucosa. Here's Pseudomonas growing in CF sputum. That's a rod-shaped bug. This is a TEM, so it's cut cross-section. It's surrounded by, these are all Pseudomonas surrounded by this dense matrix, okay, growing together in clusters. Here's an example, again, of this clustered bacterial growth in bacterial endocarditis. Here's the heart valve. This is a blow-up of this area. See a bunch of bacteria growing in aggregate clusters grouped together. Here's a chronic wound infection. This is work done by Phil Fleckman and John Olerud here at UW in a diabetic mouse model. And what you see is this is the air, the wound is way down here, this is the scab or the exudate, a bunch of neutrophils, but they're surrounding pockets or aggregates of bacteria that you can see in this blow-up. There's the bacterial clusters or aggregates. It's a common feature in most chronic infection sites, bacterial aggregation. So why does this matter? This matters for the following reason. If I take my bacterial plate, pick a colony, grow it up in liquid, hit it with tobramycin, y-axis is plotted number of bugs on a log scale. What I can see is I can eliminate them very easily with low concentrations of antibiotics. If I take the same bug, genetically identical, same resistance phenotype, grow it in an aggregate, now same graph, y-axis is bacterial number, I can go to 100 times more, 1,000 times more. I still can't eradicate this organism when it's growing as an aggregate. The key point I want to make here is that this is a phenotype. If I take the bug out of here and I grow it like this, it'll become sensitive again. If I take this bug out of here, grow it like this, it becomes resistant. It's not fixed mutations, fixed resistance caused by genetic mutations. It's phenotypic resistance. It depends upon this growth mode being in the cluster. So why is that? Why does clustering of bugs or aggregation produce such marked antibiotic resistance? I should say tolerance, antibiotic tolerance to differentiate from mutational resistance. But I always slip up on that. So there's two main hypotheses that I just want to review for you. This is not our work. One idea is that the simplest idea is that the antibiotics fail to penetrate these aggregates and don't get to the bacteria. The second idea is that the bacteria are starved for nutrients, and starvation produces resistance. We've been working on this, but I want to first show you some data on this that was done by a colleague at Montana State, Phil Stewart, who's actually an engineer. And he did this very simple experiment, which I think was very informative and changed the field, actually. And so he grew up, this is actually a Klebsiella biofilm, but he used, think of it just as a bacterial aggregate. He grew it up on agar. He had his complex sieve system with just a paper filter. And what he could do then is after it had grown up on the filter, he could lift it up. He could now put it on another agar with antibiotics. And then he could put another piece of filter on the top of this thing, and he could measure the antibiotic penetration through this bacterial aggregate to test the hypothesis that the drug just wasn't getting in there. And so this is what Phil and others have found. So on the y-axis is plotted the concentration in the top disk divided by what was in the agar. So 1 would be perfect diffusion. X-axis is time. And what you see is that the antibiotic actually gets in there quite well. And over time, you get very high concentrations of antibiotic diffusing through this biofilm matrix. So that work and others suggest that the penetration or diffusion of antibiotic may not be the most important factor in the resistance of these aggregates, tolerance of these aggregates. And that makes sense. This aggregate matrix is mostly water and polysaccharides, and antibiotics can fully penetrate these things. And if you think about these aggregates, they're even resistant to hydrogen peroxide or bleach, which should have very low diffusion effects, almost not affected by diffusion limitation. And so it's probably not this issue of penetration. So the second idea about why these things are so hard to kill relates to nutrient starvation. And it's been well known that starved bacteria, slow-growing starved bacteria, become highly tolerant to killing. And I'm going to show you another piece of Phil's work, and you can see why you need an engineer for this kind of stuff. And so Phil, I think, was one of the first to show this idea of nutrient starvation. So what he did was he did a simple experiment, for him anyway, where he took an oxygen electrode and he put it down into a bacterial cluster, and he measured oxygen tensions inside the biofilm or cluster. And this is what he found. On the y-axis is oxygen concentration, on the x-axis is depth into this cluster. And what he saw was that when he got into this thing, he didn't have to go very deep, the oxygen level started to plummet. Okay? So I already told you that antibiotics penetrate really well. Why wouldn't oxygen penetrate really well? Should even be better. And it does penetrate. But this is not oxygen limitation due to failure to penetrate. This is oxygen limitation due to consumption. What we call in the lab the buffet line hypothesis. The guys on the outside eat first, everybody else has to eat later. And you have nutrient limitation, oxygen limitation, because of the structure, the layered structure of the biofilm or the aggregate. Nutrient consumption produces nutrient deprivation at the center. So what I've told you is that these gradients result from nutrient consumption, and that we've known for many years that nutrient-limited cells resist killing by many types of antimicrobials, and that's a phenotype. It's not a genotype. If you give them nutrients back, they start becoming sensitive. And so here's just a picture of tobramycin-treated bacterial cluster from our lab. And so what you're looking at is a giant cluster of bugs. This is right here the size of one Pseudomonas. Okay, so this is a big lab-generated cluster. And we treated it with tobramycin and then treated it with a dye that stains the red cells dead and the green cells live. And what you see is a rim of killing on the outside of this cluster, with the cells inside the cluster remaining viable, which also goes along with this notion, that pattern of killing goes along with the notion of nutrient consumption.
What stimulus changes cells with bacteria from free swimming to aggregate if it's not a genetic change? Mm-hmm. What tells them to change their lifestyle?
That's a really good question. And that's a controversial point in the field right now. We know that in certain laboratory reactor systems, I can genetically mutate bugs, knock out certain functions, and they can no longer produce biofilms or aggregates, suggesting that certain functions are required to build these things, self-assembly, it's development. However, in chronic infection sites, I don't think that that's what's going on. I think that bacteria may aggregate by passive mechanisms, either due to the viscosity of the medium, for example, wound fluid or mucus. You could imagine just simple growth could produce a clonal cluster of bacteria without any active process going on, just because of confinement. in a thick medium. And so I think it could be very active in some circumstances. I think it could be passive in other circumstances, just based on the growth conditions. And that's an important distinction, because if it's active, then we need to target those functions in treatments. If it's passive, it's going to be more difficult, just a consequence of where they're growing. So, let's see what time we got here. Okay. So, what I've shown you so far is that CF and other chronic infections, we see bacteria living in these clusters or aggregates, and that nutrient consumption by the peripherally located cells causes starvation in the central regions of cell clusters, and that contributes to antibiotic tolerance in these things. So I'm going to tell you just about briefly about a couple of strategies that we're using in the lab. Maybe I'll just get to one of them to try to figure out how we can sensitize starved bacteria to killing, because that's a key problem. And so the first question really is, why is it that starvation produces such marked tolerance? And the conventional explanation is that well, your antibiotics, just like your chemotherapeutic agents, target growth and replication functions. You target DNA synthesis, cell wall synthesis, RNA synthesis. And if you're not growing, you're not using these targets. Or if you're starved, you're not using these targets. And if you're not using the targets, the anti-target drug has reduced efficacy. So what this idea says that nutrient limitation reduces growth and the availability and the importance of the targets that we're going after with our drugs. A different idea is not that it says that slow growth and starvation are not inherently linked, but rather the idea here is that the bugs can sense nutrient limitation. We know they can, because they respond to low nutrients by turning up acquisition systems. They sense nutrient limitation, and that sensing turns on responses that produce phenotypic resistance. This is a passive mechanism. This is an active mechanism. I notice that nutrients are getting scarce in the environment. I turn on functions that make me tolerant. So that's the two general big picture possibilities. And so let me show you about one experiment that we've been doing to try to distinguish between those possibilities. So before I can do that, I got to tell you a little bit about how bacteria sense nutrient starvation. And so basically, a very conserved mechanism is something called the stringent response. And so what happens is, here's my mRNA, here's my ribosome, it's going along, it's translating RNA into protein, and I develop amino acid starvation. And through mechanisms that are not important for this talk, when the ribosome senses that there are not enough amino acids to hook on to the growing protein chain, it turns on this starvation signal, which is a guanine tetraphosphate molecule. And that starvation signal diffuses throughout the cell and tells the bacteria, things are getting bad. We're about to hit serious nutrient starvation. Prepare for this. That's the nutrient starvation system in bacteria. And so upon starvation, this alarm mode, we call it, modulates gene transcriptions to prepare the cells. So one thing I got to tell you to understand the experiment I'd like to show you is that we can mimic this in the lab by putting in an analog of amino acids that cannot be incorporated into protein. So it's defective. It's a serine analog. It can't be added to growing protein changes. The cell senses that as starvation because it's out of serine, clogs up the machinery, and it turns on this alarm signal. So it's a trick for us to use in the lab to turn on and produce immediate starvation by gumming up the works with the serine analog. So we mutated this stringent response. The bugs, these are bugs now that can't sense starvation. And I want to show you one experiment. Actually, two experiments. So the hypothesis is that the resistance to antibiotic killing requires that the bugs sense starvation and then actively respond to it. So here's the experiment. I measured the killing of wild-type bugs that can sense starvation fine, and these starvation-sensing mutants when they're nutrient limited to see what the difference was. So here's the experiment. Here's my growth. This is optical density, a measure of bacterial growth. There's time on the x-axis. Time on the x-axis. And this is what I normally see. Bacteria have a little lag phase and they start growing. When I add my serine analog, I get starved cells, and growth immediately stops. Now, this happens in the wild type that can sense and in the mutant that can't sense, right? If your gas gauge is busted, you can still run out of gas. You're just unaware that you've run out of gas. So in the wild type bug, they're aware of it. They can sense starvation and respond. The mutant bugs can't sense, but they still starve. And then I apply an antibiotic and I look at the difference. It's a little bit of a complicated experiment. But let me walk you through the data. So this is the wild type bacteria. These can sense starvation just fine. Y-axis is plotted the number of bugs. This is no untreated condition. Okay? Got a lot of bugs. Here's, I add antibiotics, ofloxacin. I reduce the bacterial counts by 3-4 logs. A good killing effect. Now I add my serine analog and ofloxacin. I've starved them, plus they know it, okay, and they've become highly tolerant to ofloxacin. That's the expected result. Now, what happens if I do the same experiment with my starvation-sensing bacterial mutant? Similar number of cells. I put ofloxacin. They're actually a little more sensitive to ofloxacin alone. But now when I starve them and hit them with ofloxacin, they don't develop the full resistance phenotype of the wild type. So what this data says, and other experiments that we've done, is that starvation isn't enough. They have to sense starvation, respond to it, and that produces the full phenotypic antibiotic resistance. So that's a very hopeful finding, I think, because it tells us that maybe we can target this sensing system to sensitize starved bugs. So this is just one last experiment. I'll show you that this same effect occurs. In that other experiment I showed you, I produced an artificial starvation by putting in that serine analog that gummed up the words. Here I produce starvation by growing the bacteria in aggregates or biofilms, where the starvation is occurring naturally by the metabolic activity of those peripheral cells. And I see the same phenomenon. The wild type here is no antibiotic. The wild type is highly tolerant to all these different antibiotics when it's grown in a biofilm. Starvation sensing mutant, similar number of cells, a little lower, it becomes much more sensitive to killing by these different antibiotics. So I think I'm going to end there because the other stuff I had required some movies which aren't going to work. So I'll just conclude then. The data I've shown you or the ideas we're thinking about is that genetic diversity in infecting populations provide a kind of biological insurance, increase the population's stress resistance. Bacteria live in these biofilm-like cell aggregates at the sites of many kinds of infections, and these are inherently resistant to killing just by being in the aggregate. Aggregates resist killing primarily or largely because of nutrient-limited subpopulations in the center of these things. And nutrient-limited subpopulations can be sensitized by blocking the ability to sense starvation. I didn't get to show you this because it involved movies. I can also sensitize these bugs by turning on genes that induce the bugs to disperse, to get out of those clusters. So I've shown you work by a lot of people in the lab and some in other labs, the work by Phil on the penetration and oxygen. He's at Montana State James Palmer gave me that slide of Sinus biofilm or aggregate, and Sottinger did the diversity work, and others in the lab contributed to as well. So thanks a lot for your time.
So is there anything we can do now to improve treatment outcomes, translate this into clinical practice?
Well, that's a good question. I think that a very simple thing that one could do is to use antibiotics in combinations. And I think that's particularly important for chronic infections because of that diversity effect. If you think about, you know, if the mutation frequency any given of a bacterial population to any given antibiotic is, say, 1 out of 10 to the 9th bugs is going to be spontaneously resistant to any antibiotic you choose. So if you just give me a clone, I grow it up to 10 to the 9th, which is very easy to do. It takes about 8 hours or less. Okay, there's going to be a spontaneous mutant in every gene in any given gene in that entire genome. There'll be some mutant in there. So 1 out of 10 to the 9. Now, if I put 2 antibiotics simultaneously, right, it becomes 1 out of 10 to the 18 to be doubly resistant. The probability is the product of those individual mutation rates. So I would say, my own feeling would be that would be the simplest thing, using antibiotics in combination when possible. The other stuff is years away.
So, I mean, if you've tested that, that's obviously the model of TB and HIV and all these other places, but here it seems a different mechanism. I mean, going back to your 96 well plates, you have wells with 1 antibiotic, 2 in combination, 3, 4, 5, 6, and do you ever reach a point at which you say, all right, nobody has a bug resistant to these 4 antibiotics?
Yeah, that's a cool experiment. Do you want to do it? I think that's a cool experiment. We haven't done that. You know, we're actually trying to get away from the phenotyping binning because it's driving people crazy. And no one is really harmed in the process of those experiments, but they're scarred because there's tons of replicate cleaning. But I think that's a nice idea. What we're moving towards now is trying to do whole population genome sequencing. So take 1,000 bugs that coexist in the patient, 1,000 clonally related siblings that coexist in the patient, pool them, sequence them, measure allele frequency. Say, okay, he's well. What's the frequency of certain alleles in that population? Now he's sick. What changes? You know, could it be that the disease? The other thing about chronic infections is that we all have disease flares. What's going on in a disease flare? We got a CF patient. They had 10 to the 9th Pseudomonas per millisputum. They're well. 2 weeks later, they have 10 to the 9th Pseudomonas per millisputum. They're sick. How does that make any sense? Could it be that there's flares or blooms of more invasive variants during that? We're trying to look at those.
That seems to be the best way to get at the true diversity. The problem is you'll lose the functional. I mean, you think from just the genes you see, you'll be able to get at function and resistance?
It'll add additional steps because then we'll have to engineer those mutations into laboratory strains and test it. That's right. That's exactly right.
What type of existing drugs target these nutrient-resistant organisms? Are there some that can be exploited, or
that's a very good question. There is literature that suggests, this is all for Pseudomonas, so wouldn't generalize it necessarily, but there is literature to suggest that quinolones and antimicrobial peptides like polymyxin or colistin are less susceptible to inhibitory effects due to slow growth, and particularly the polymyxin and colistin class, because those act by permeabilizing the membrane, putting a channel in the membrane of the body, releasing transepithelial or transmembrane polarization, putting a hole basically. And you could imagine that if you're putting a hole, you don't have to be necessarily growing. It's not targeting DNA synthesis or ribosome action. And interesting, most host natural antimicrobial peptides in your tears or saliva or sputum work by that mechanism, permeabilization. So I wouldn't give any clinical advice, but that's what's
And that alarmone, can that be targeted?
Yeah, we're working on that. I think that could have some promise. And so, a former postdoc of mine is working with a company to do high throughput screening of compounds that inhibit that process, because it's conserved in gram-positive, gram-negative bacteria all throughout. So that's possible.
So, given all the work you have here, do you put much? I mean, the clinical microbiology testing we do now and
Yep.
sensitivities, does that mean anything?
Yeah, that's wonderful. I don't know the answer to that. The European CF Foundation is actually issued guidelines saying don't do susceptibility testing anymore. Not because of these findings, but just because it doesn't work. It doesn't work very well. In chronic infection, in chronic CF infection, I think in acute infections the data is quite good, right? You know the MIC, it helps you guide, produce the bug. In a chronic infection, my own feeling is it probably doesn't work very well. And in CF, what we often do is we ask the patient, what works well for you? And we send the bug, we get a bunch of R's and S's, but I don't know if it's useful to use it.
You have to realize, though, for young people here, the Kirby-Bauer technique of bacterial resistance was developed here at the University of Washington. So saying it's not a good test is silly.
I qualified it. I said chronic infection under some circumstances, but I appreciate it.
If you use combination antibiotics, as you were suggesting before, aren't you simply going to increase the genetic pressure to develop other modes of resistance?
Could be, yeah, it could be. I mean, I think that it's a numbers game. If it's really, say for example, I can use enough antibiotics to get the probability of a triply or quadruply resistant mutant up to 1 to the minus 36, maybe I'm okay. I don't know. The other thing that we know about CF is that, and I don't know if this is true in chronic infections or not, but in sinus infections, but in CF isolates, many of them have developed mutations in mismatch repair genes that improve DNA replication fidelity. So they have higher mutation frequencies or mutators than the initial strain. So if you throw a mutator strain in the mix that has 100 or 1,000-fold higher mutation frequency, I don't know what's going to.
Well, thanks very much.
Thanks.
Yeah, but it's just like what they were saying. Yeah, because it was only a few that were, from they weren't created activity.
I've been out of town, but I'll get
So apparently he didn't get enough blood at one time. So we called him back. I really did. So I called him back, but then
Everything else is good?
Oh, validated. No, it's fine. I know who could help you, but
Okay, well that's yes. She's got such an unusual.
Yeah.
Do we have how much
But no, I think that's a good. Like, that's for the. Yeah, they were talking about that.